Good afternoon, guys. Today we are going to continue with part 2 of lecture 1 of chapter 1. And we're going to move into talking briefly about some morphological concepts that we're going to use in lab, and then talking about phylogeny so we can get ready to go through the taxonomy and phylogeny of organisms so that we can then move into comparative anatomy. So let's jump into it. So comparing structures. The two ways to compare structures really are thinking of structures that are called homologous structures and structures that are called analogous structures. Homologous and analogous structures often look alike, and that makes it hard to determine whether, in some cases, that there's actually some ancestral or some phylogenetic significance to that. That is, if these organisms share common ancestry or not. So comparative morphology, again, study of similarities and differences among species. And similarities may be considered in several different ways here. The first one are homologous structures. This is when two or more structures have similar parts or the same parts, maybe the same bones, and they share common ancestry. So when you look across here again, I showed you the limbs, four limbs, right, of amniotes. We have a human, cat, frog. Well, frogs are amniotes, bats, and birds here. They all have the same bones. And so these tetrapods share a common ancestor, most likely because they have the same bones, right? And you can actually trace these back to common ancestry based on their genetics and the fossil record. So homologous structures, two or more structures that look alike, two femurs, for example, in a cat and human, and they share common ancestry. Homologous structures are the kinds of structures we're trying to use when we are doing phylogenetic trees, when we're building trees on ancestry, as you'll see here in a few minutes. Analogous structures are body parts that perform a similar function and often look alike, at least externally, but there is no common ancestry. So wings are a great example of this, right? Insect wings versus bird wings. They kind of look alike on the outside. On the inside, they look completely different, most likely because they don't share common ancestry at all and were built differently. So these are called analogous structures. Often structures that are analogous happen because of something called convergence, which you see down here on the bottom of the slide. And it's the environment that often shapes structures to look alike, even though common ancestry is not shared. And this is called homoplasy. So homoplasy is often caused by convergence when two things look alike. So analogous structures are two or more structures that could have similar function but different structures, or they could have similar structure and different function depending on what's going on. And they're only seen often you see differences internally. And these are analogous structures. Many analogous structures are homoplastic, which means they were caused by environmental constraint. So these kind of two terms kind of go together, homoplasy and analogous. So homoplastic structures, two structures that are apparent similar and often do the same thing within the environment, but we do not share common ancestry. One other thing I'll add is analogous structures, if you do add them to a phylogenetic tree, can really mess things up, right? Because you're basically putting two animals in a group, or in what we're going to call a taxa. A clade is the name we use, as you'll see in a few minutes, that don't belong together because they actually don't share common ancestry. This is one of the places where traditional taxonomy, or what we call classical taxonomy, differs from phylogenetics. In classical taxonomy, we base our group animals based on similarities, so we don't care much, or at least classically, these taxonomists didn't care much whether they were related evolutionarily or not. Today, we're trying to fix those classical taxonomic schemes so that taxonomy and phylogeny are similar, but it's not always that easy. So analogous structures are very useful in taxonomy. They're also very useful to know so that we don't include them in phylogenetic trees where common ancestry is not shared. So you can imagine convergence is often what can muddle, if you will, or mess up phylogenetic trees. So organisms evolve in environments in similar ways. Wings and fins are two great examples of structures that are formed in more than one taxonomic group that don't share common ancestors. So fish and dolphins, for example, don't share common ancestors, right? And this is often called convergent evolution, and it's very useful in our understanding of how organisms have evolved over time. But convergence can also mess up phylogenetic trees when we try to put them together if we don't recognize them. Okay, let's talk a little bit about symmetry and then some directional terms before we move into talking about what phylogenetics means. So there are definitely different body plans that are found out there in the animal world. The two major body plans that you see are called radial symmetry, and the other one is called bilateral symmetry. Radial symmetry is very common in organisms that don't move very well. A lot of invertebrate groups have radial symmetry because they don't move to the environment very efficiently. And so if you have radial symmetry, you basically surround your entire body structure around a central axis. Think of starfish, think of jellyfish. And in this way, they can interact with their environment on 360 degrees and gives them higher probability of running into things because they don't move well. On the other hand, bilateral symmetry are found in organisms that move quite well through the environment, and all vertebrates, the group we're going to study, are bilateral, which means if you take a saw and cut right down the middle of the organism, you get mirror right and left halves, and you have kind of a cephalic or head region and then a tail region, or towards the feet region, if you will, in organisms. So a mid-sagittal plane is a cut that would give you equal right and left halves in the organism. And so animals that take on bilateral symmetry actually move through the environment really effectively because of their body form or their body axis. Now, there are also directional terms on this slide that are going to be really important in the lab. And you need to become really, really familiar with these so that we can use them in the lab. You're going to see them in the lab handout. I want you to use them with your lab partners. And when we're talking about not only that there are certain organs in the body, but how the organs are situated in three-dimensional space. And that's what directional terms allow. And so here are kind of the main directional terms here on this slide and the next slide. And notice with animals, some animals like rodents can be bilateral in certain situations, and then quadrilateral in others, are what we call quadrupedal and bipedal. You look at our surfaces, if you will. Let's do the quadrupedal organism first, because that's what we'll deal with a lot in the lab. We have what's called anterior, posterior, dorsal, and ventral. If you're looking at a quadruped, this is what it looks like. The dorsal surface is the back. So that's the back of the body. The ventral surface is the belly. All right, so dorsal and ventral. And then towards the head is going to be called anterior, and towards the tail is going to be called posterior. I don't like anterior and posterior because they're also used with humans and other primates. Notice ventral and dorsal. In a human, they like to use posterior and anterior. And with the humans, they use superior and inferior. Again, terms that I don't use a lot in comparative anatomy because we're not studying humans, we're studying other organisms. So we will save posterior and anterior and superior and inferior for your human or more mammalian kind of classes that you might take in anatomy. So for example, if you were to take gross anatomy, you would use those terms. We're going to stick with dorsal ventral for our back and belly, and then cranial, head, caudal towards the tail. That's what we're going to use. Notice as the animal stands up, just cranial and posterior are going to be up, and dorsal and ventral are going to be facing forward and backwards, right? Now, if you cut the organism along these directional planes, then you can get sections. And you can see over here we have a whale showing you the different views if you were to take photographs, right? So you got basically an anterior view here, what we call cranial view, and then we have a posterior view with a tail view, or what we would call a caudal view. This is the ventral view of the belly and the dorsal view of the back of the humpback whale, right? We can also cut that whale using sectioning, right? Sectioning. And this sectioning becomes really important on the whole organism, or if you're thinking about an organ. If I take a heart out and I section it in different ways, I'm going to get different planes of view, which give me different information, right? So these are the major sections that you can see in textbooks and during dissection. Sagittal sections cut a organism into right and left halves. Mid-sagittal section cuts the right and left half into equal sections. A parasagittal would be off to the right or left and give you unequal sections. A frontal plane is one that cuts kind of through the body so that you get a dorsal and a ventral half. All right, so if you get a dorsal or ventral half, so if you take a saw and cut down this way and you pull the front of me off so you can look inside, right, that would be kind of what we would call a frontal section. A transverse section or a coronal section cuts things in cross sections. All right, it gives you a cranial and a caudal end, right? So you can see that if I were to cut through the head this way and I pull the cranial part of my head off and I could look down into it, that would be a cross section, all right, or a coronal section. Notice these sections, these major ones, we try to do at right angles, right? Because that gives us the best information. If you were to section at an angle that other than 90 degrees, it gives you really strange looking sections. So if you ever get onto a microscope and you see an organ that looks really strange under the microscope, it wasn't cut directly at 90 degrees in one of these planes to give you the view of interest that we're interested in seeing. So those are the planes and some of the directional terms. There are a few other directional terms we need to learn about and a few other areas. So we can combine directional terms, as you can see over here with the cat, where, you know, we can move dorsally up or we can move ventrally down. But what if we move at an angle to that? So what if we move from dorsal to ventral, but we're moving towards the caudal end? So we could call that posteroventral or caudal-ventral, right? Caudal meaning moving back towards the belly, or you can move up towards the head, and you could call that anterodorsal or craniodorsal, right? As you're moving towards the back, you are actually moving forward. So you can combine terms. Here's are a couple of others down here you could use, where they're using posterior-medial or anterolateral. Again, you could throw in there cranial and caudal, right? So as far as moving towards the tail or towards cranial, towards the head. Laterally, you'd be moving out towards the limbs. Medial, you would be moving in towards the belly button. The other major directional term that we're going to want to use is distal and proximal. All right, so this has to do typically with the limbs. All right, and so when I put my limbs out, if something is distally located, it's located further away from the trunk. So my fingers, right, my fingers are distal to the rest of my forearm, right? They're furthest away from the trunk. Something that's proximal, like the shoulder joint, is much more proximal to the trunk, closer to the trunk than the fingers. And that's how you would use distal and proximal. Two other regions we'll talk a ton about when we talk about vertebrates are the pectoral region and the pelvic region. Pelvic means hips, so we're talking about the hip here, hip area. Pectoral means chest, which would be the chest region. So those are two areas where we'll look a lot at, particularly when we get to the development of the limbs. So I love this slide here. This slide gives you basically all of the terms kind of on the same page, if you will, right? So again, anterior towards the head, we use anterior cranial, right? Superior is only in humans. Posterior is towards the tail. I like to use caudal. Inferior is only in humans. So you can basically X out superior and inferior, right? We're not going to use these terms in this class because those are human terms, right? But we can use cranial and anterior interchangeably. We can use posterior and caudal interchangeably. Dorsal is what we're going to use for the back. Again, we're not going to use posterior, and I'm really not going to use anterior for belly, right? We can use anterior so it's not confusing as only towards the head. So we're going to use ventral for back and ventral for belly and dorsal for back. Medial means towards the midline, towards the middle of the organism. Lateral means more towards the outside of the organism. Proximal, closer to attachment. Distal, further away. Superficial is when it's closer to the surface. So the skin is the superficial to the muscle underneath. The muscle's deep to the skin, right? It's further in than the skin is. So those are two other terms that we can use histologically or to think about three-dimensional depth. All right, I think that does it for directional terms. Again, get used to using these in lab. You'll see them on the test. You'll see them a lot on lab exams. You'll see them in lab handouts. All right, one other major type of organization that you see within organisms is called metamerism, or what's called segmentation. Parts of bodies can be broken down into segments. In vertebrates, segmentation is very, very common. Think of the insect segments like the head, the thorax, the abdomen, or if you think of a segmented worm like a tapeworm is made up of segments. Segments are much more rare in vertebrates. In the adult, where you see segmentation most is with the vertebrae, right? The vertebrae kind of all look alike, or maybe the ribs you can even think about as being segmented, but it's really the vertebrae. You can also see segmentation in vertebrates in development, like the somites, which are balls of developing connective tissue or mesenchyme that all are about the same size that line up on either side of this tube right here in the middle. The tube right in the middle is the developing spinal cord. It's another instance of segmentation. Of course, those somites disappear as those cells migrate and form all of the connective tissues of the body. But that's another kind of organization to the body that I thought I'd mention because you run into it with vertebrates. Okay, one other interesting at least structural aspect we should talk about are what are called vestigial structures, because we'll run into them as we compare different organisms along the way, different vertebrates through dissection and through lecture. Vestigial structures are structures that are basically remnants of what they used to be because they're not really used anymore as far as the adaptation of the extant, what we call living organism. So if you look up here, this is one of the ancestral fossils of the cetaceans, of what we're going to call later on the dolphins, the whales, the porpoises, right? And you can see this animal has limbs because it actually is going to have arisen from a group of land mammals. And so it does have limbs early on in the development as it moves into water. This is the ancestor of the modern whales. And if you look at a modern whale or a modern porpoise today, they still have remnants of their hind limb, right? That you can see there. That is called a vestigial pelvis. And that's all that's really left over. You see this in snakes too. There are several snake species, particularly those that we consider more primitive or basal. They have remnants of the pelvic girdles, even though they do not have lower limbs, right? And these are called vestigial structures. So there's structures that are left over from the evolutionary history of the organism that aren't in use today. All right, so that leads us into now we can focus the rest of the lecture on classification and phylogeny. We're going to do a comparison of what we call taxonomic classification versus phylogenetic classification. You've got a little of this in your hopefully in your biology 155 class. We're going to use both taxonomic and phylogenetic schemes. Sometimes I even intermingle them as there are taxonomists and biologists, evolutionary biologists that are trying to combine the two fields, if you will, to taxonomy and phylogeny. So let's dive into it. Okay, so let's start with classification. Let's start talking about classification here. All right, so one of the earliest ways that we started classifying organisms is based on what's called binomial nomenclature, which was invented by Carlos Linnaeus in the 1700s. And what he tried to do is start grouping animals based on their similarities. And he started giving them two-part names. And this two-part name system has stuck. If I were to find a new beetle today, the first thing I would want to do is give it its binomial name and publish that. And that would be its genus and species name. And that's what Linnaeus did. The genus name is the first part of the name. The species name is the second part of the name. And basically each animal that we have described, whether it's a vertebrate or not, has been given a binomial name. And so over here are two examples of this, where we got examples of two cartilaginous fish here. We call them rays. Here's the manta ray, and here is the concentric ray. You can see manta is the genus name and Hamiltonii is the species name. I want you to notice that the scientific name, the binomial nomenclature name, is italicized, and the genus is always capitalized, and the species is always lowercase. All right, and we do this naming and grouping to make the organisms easier to study, right? It's really hard to think of vertebrates as a group. I mean, it's daunting. There are lots of vertebrates. It's much easier to break them down and think about birds and reptiles and mammals and frogs and salamanders, right? And you can actually, as a scientist, go in and study one particular group, right? Makes it a lot more digestible, if you will, or a lot easier to study them that way. So zoologists have been, grouping vertebrates and other animals since Carlos Linnaeus, who came up with binomial nomenclature. Now, others have added other taxonomic groupings, and we kind of done it in a hierarchical fashion, right? So the most specific name is the genus and species name at the bottom here, right? So every organism gets its binomial name, which is in italics. It's italics, right? Italicized here. So in this particular example, we're looking at this frog, one of my favorite frogs. This group here are called dart frogs or arrow poison frogs, and this particular genus is called Dendrobates. And dendrobatid frogs, the specific frog here is called Tincturus, which means painted one on the back, and you can see the nice yellow and blue here. So that's the genus and species name. And what scientists and biologists, zoologists are interested in, taxonomists in particular, is then figuring out what group that particular frog belongs in, right? And so it's going to put it into a larger group, and then that group's going to be in a larger group, and a larger group, and a larger group, until you get to the largest kind of taxonomic grade, if you will, which is called the kingdom, right? You guys have seen this before. So here are the taxonomic hierarchical levels or grades, if you will. They are called kingdom, phylum, class, order, family, genus, species, right? And for a long time, taxonomists simply put animals in these groups based on their similar structure, what they look like, right? And often that works out really nicely because structure is often determined by common ancestry, but that's not always the case because of convergence, right? Which is something we've already talked about. But in any case, if you look at this frog, Dendrobates tincturus, it actually belongs in a family of frogs called Dendrobatidae, which are all tropical frogs, South American and Central American frogs that all belong together. Many of them are brightly colored and produce toxins on their skin or poisons on their skin. This family of frog belongs in an order of all the frogs, which is called Anurin. So every frog is found in the Anurin order. Anura means tailless one. And so all the frogs are found in here. There are lots of families of frogs. Dendrobatidae is one of those families. What I want you to notice are most family names and animals end in DAE. So you know that's a family name if you see the DAE. And frogs are part of a larger group called amphibians, right? Modern amphibians, and that's the class Amphibia or Lis Amphibia. And amphibians are one group of what we call vertebrates, right? Which is the subphylum here, right? And vertebrates belong to a larger grouping called a phylum, which is Chordata, right? And then all chordates are found in the large group we call Animalia, which includes all of the other invertebrates that are found out there in the world that we consider animals. All right, so that's kind of the most generic grouping there, the kingdom. It's also the oldest group, right? All animals, right, are found in the kingdom. And hopefully, if we can build phylogenies off this, as we go down, these groups get younger and younger and younger, so that the genus is the youngest or the most recent division. If we can do it correctly on a phylogenetic tree, it's not always possible, at least right now, right? So that's taxonomy, all right? And we'll look at different taxonomic schemes as we go along. We'll also look at phylogenetic trees, which don't always follow this taxonomic setup. So with phylogenies, with phylogenetic trees, which I know you've seen a lot of, what biologists are doing there are trying to group animals based on their ancestry. Using similar characteristics is great, but it's not always useful, is it? Because of convergence. So there are groups of animals that look nothing alike that actually share common ancestry. And so that's really, really important. And so, you can see different types of setups here with phylogeny, and some of them can be quite dangerous. So this setup you see here, which is listed in your textbook, is also one I've seen in a non-majors book. What it shows is that these animals are related to each other. Really what it assumes is this animal gives rise to this animal, which gives rise to this animal, that gives rise to that animal, and that's not the way it is. These are the animals on the top of a phylogenetic tree, like you see over here. So this animal would be F, this animal would be A, this animal would be R, and this animal would be B, and this animal would be M. And you notice they don't give rise to each other. You have to trace back a long way to form a common ancestor between all of them, right? Each one of these lines is an independent line of evolution that's been evolving over millions of years, independent of all the other groups. So these two really are not closely related. You have to trace way far back to find a common ancestor. A lot of textbooks, or you'll see in some writing, call animals lower and higher, which is a terrible term because animals that are down here on the phylogenetic tree are not necessarily lower than the animals that are up here. That is, fish aren't lower than mammals. They just belong to a different line of evolution, right? And so the animals that you see today at the top of a tree, all of them come from independent lineages, and each of them are really, really derived or very, very complicated animals compared to their ancestry, right? Or their oldest ancestors. So I like to use primitive and derived. Or I like another term instead of primitive that I like to use is basal. I love the term basal, or even ancestral is another one that we like to use, which are better terms that tell you that they're closer to the base or closer to the ancestor of the organism than derived, right? So derived is another term. Derived, just if you think about the word derived, means that they're a little more advanced or have different characters, if you will, than the groups that are below them. So this is a bad way to represent phylogeny. This one here's a better way. This ladder effect is definitely not a way to do it, right? So we want to X that out, and we don't want to do that, right? This is, a much better representation of the evolutionary ancestry or history of organisms. So again, we looked at classification, and there's a hierarchical scheme to it, right? And I like this figure a lot also. So it kind of shows you this is a class of animals, right? So remember, we have kingdom, phylum, and class, right? So we're looking here, this is a class of animals, and within that class, there are two orders, right? Which are smaller groups of organisms within that class. And within that class, within this order, you have two families. In this order, you have one, and you have 3 genera here and 4 genera here, and then you got all of these different species. And this kind of shows you how you can go down and down and down and down until you get to the individual species. So within a genus, these species look very similar or share very similar characteristics, whether that's genetic characteristics or morphological characteristics often. There are some genera where the species look nothing alike, but their genetics is very similar, right? And that's because they are niche specialists. They live in niches that are so different from one another that it's really, really tailored what they look like to the niche and not necessarily to their ancestry. Does that make sense? So this is the hierarchical scheme, and often phylogeny doesn't follow it, right? Doesn't follow it completely or as closely than you see here, right? And that's because phylogeny is more interested in evolutionary relationships, not necessarily in similarity. So again, here's your hierarchical classification: class, orders, families, and genus, right? Trying to group them based on similarities and where they belong. And here is the phylogeny, right? Now, what we're trying to do today is match these two up. So, a lot of times what we're trying to do now is fix the taxonomy so that it sits well with the phylogeny. And it's not always an easy thing to do. But you can see here we kind of work the phylogeny to match what you see in this particular setup. So the only objective for classification really is the evolutionary history, regardless of what the organisms look like. And that's really what we're trying to do with the Tree of Life, which there's a huge website online called thetreeoflife.org, which is trying to map out, if you will, the evolutionary relationships between all life. So it's treeoflife.org. You can look that up. It's a pretty cool website. You can get lost on it, Tree of Life. And there you can look at a lot of what we now believe are the evolutionary relationships, at least presently, of a lot of the organisms. And not all of them have been resolved. We don't have the right fossils, or the animals that are alive today we haven't collected genetic information from all of them. Okay, so phylogenetic trees is a diagram that describes the phylogeny or the evolutionary relationships between groups of organisms. And it is a hypothesis. That's the first thing I always teach. It's a hypothesis, which means it might not be correct in 3 years. And 3 years ago, the phylogeny isn't correct of what we see today. It's based on the available information we have today and is the hypothesis of the evolutionary relationships among various species. If we add more species to the list or we find something that changes it, then we will publish that and we will fix the phylogeny. So phylogenies are always changing. And that's what the general public, particularly those that believe in creationism, like to attack evolution, that we never know what we're talking about. It's because they're not using the scientific method like we are to describe what we understand today. All hypotheses are educational guess on what we think is right based on the information that we have presently. New species can be formed in two major ways, at least that's how we've described it so far as evolution goes. They can arise from anagenesis, where a single species gives rise to different species. That is, one species kind of gives rise or turns into the other one. That is, one species gives rise to another one and then probably dies off, right? And so C gave rise to D. Or more commonly by cladogenesis, where a species diverges into two, right? Two species. Okay, so this is what a phylogeny looks like. It's based on lines. We have 4 species here, a simple idea, and we trace them back to common ancestors, right? And basically the diagram will show branches off of a line, right? And this line represents a time scale typically. And if it's done right, phylogeny can trace all of the species within a group back to a common ancestor, or at least a suite of characters that we think the common ancestor has if we don't have the fossil for it. Here's another way to represent the same thing, is using this kind of branching technique, and the common ancestor would be down here, right? D would be your outgroup in this example because it is not in this particular clade, right? Or would be very basal. So you can see here we're using D to define A, B, and C, or A, B, and C are next to D. A, B, and C are considered sister to D, if that's what is the term that they often use when groups are close to each other or right next to each other, sister to each other. So for example, A and B are sister to each other, right? And A and B are sister to C, right? And then A, B, and C are sister to D, right? You see that? That's how it kind of works. So you can see here, A and B are most closely related because they share a common ancestor. We can call it E. That C and D do not, right? So this common ancestor, they share really closely to each other. So they're more closely related to each other than they are C and D, right? And then A, B, and C, they share a common ancestor too. So they are more closely related to each other than they are to D, right? As you can see. And then the base of the tree is the common ancestor for all these groups. So basically, you've divided it down until you get all of them divided out based on recent common ancestors. It's pretty intuitive. The problem is when you get the groups get really big and you start using lots of characters to define these groups, it becomes really hard to keep track of all of them. And that's why phylogenetics often uses computers. And that's why phylogenetics has really come a long way in the last 30 years because of computers, right? The bigger the supercomputer, the faster the computer is, the more information it can process, the larger the phylogenetic trees we can do. So what are the tips and branches and lines and all that represent, right? The tips of a phylogeny are the actual living organisms, right? They're the descendants of the ancestor. So often these are extant. Extant means living organisms. So extant organisms. Extant are living, right? Extinct are dead, right? So extant versus extinct. So a lot of the ancestors that you see in the tree are extinct organisms, and then the ones that are alive today are the most recent, are the extant. So the ancestors found in the past, the descendants on top are recent, are living today. The nodes, where lines meet, represent ancestors or suites of characters that we think are ancestral. Those nodes will be defined typically in a phylogenetic tree in programs that We'll group characters. You have a backbone, you don't have a backbone. You have this base sequence, you don't have this base sequence. And that's what will be represented at the branches. The lengths of lines are often correlated to time. So the longer the line, the older the group, right? So notice number 1 is a really old group here, right? Whereas number 4 is a very young group, isn't it, compared to the others? All right, so here's an example of a phylogenetic tree, right? This is a group of birds. That includes cardinals and some of the birds you might be familiar with here. So we got some finches on one side, we have the cardinal, we have the oriole here that's also found here. And what it does is it just shows you a phylogenetic tree. And notice all these birds here are extinct birds that we got down here, right? And then the ones on top are actually living species. And you can see we can trace them back to a common ancestor. And most of what you see here is cladogenesis, where a species gives rise to 2. And then notice A gives rise to F, D gives rise to G. That's anagenesis. And then you get branching. Anagenesis happens sometimes for various reasons, which I'm not going to explain right now. But these are the living groups on top. The nodes represent common ancestors or suites of characters, right? And then you can trace everything back to a particular ancestor. That would be a nice phylogenetically sound tree. And the large group that's phylogenetically sound or evolutionarily sound, or at least we think is called a clade. A clade is a taxon of animals that we think we can trace back to a common ancestor. All of them trace back to one common ancestor. All right, I think that's where I'll stop today. And then we will pick this up talking about phylogenetic trees in the next lecture. Have a great day.